Good morning, students. Hope you all are fine and safe. Today's thought: the best preparation for tomorrow is doing your best today. In the previous session, we discussed about steps involved in recombinant DNA technology, and also we have learned the tools involved in genetic engineering process. What are the basic tools? The enzymes, vectors, and host organisms, isn't it? In that we discussed about enzymes, different kinds of enzymes, especially restriction enzymes, DNA ligase, and alkaline phosphatase. Today we shall learn about another two important tools, vectors and host organisms. First, let's discuss about vectors. What are vectors? Vectors are the delivering system of targeting gene. Vector is a vehicle which will carry your target gene into the host gene where you want to insert. The plasmids are the most suitable vectors. What are plasmids? Extrachromosomal circular DNA found in the cytoplasm of E. coli is called plasmid. The vectors are the major component of a gene cloning experiment. A vector is a small DNA molecule capable of self-replication, and it is used as a carrier and a transporter of DNA fragment which is inserted into it for cloning experiments. Vector is also called cloning vehicle or cloning DNA. Vectors are of two types. One is cloning vector and another one is expression vector. Cloning vector is used for the cloning of DNA insert inside the suitable host cell. Expression vector is used to express the DNA insert for producing specific protein inside the host. Properties of vectors. Vectors are able to replicate automatically to produce multiple copies of them along with their DNA insert in the host cell. What do you mean by DNA insert? That is the DNA of donor organism or gene of interest. In the previous class, we studied about how to prepare this insulin. In that, which is the donor or insert? That is pancreatic cell, isn't it? If it is a vector, it should be small in size and low molecular weight. Its size should be less than 10 kilo base pair so that entry or transfer into the host cell is easy. Kilo base pair is a unit to measure the length of the nucleic acids. A vector must contain an origin of replication so that it can independently replicate within the host cell. It should contain a suitable marker such as antibiotic resistance or ampicillin resistance to permit its detection in the transformed host cell. Vector should have unique target site for integration with DNA insert. Most of the commonly used cloning vectors have more than one restriction site. These are multiple cloning site or polylinga. Presence of multiple cloning site facilitates the use of restriction enzyme of choice. The following features are required to facilitate cloning into a vector. Origin of replication. This is a sequence from where replication starts, and a piece of DNA when linked to this sequence can be made to replicate within the host cells. Selectable marker. In addition to origin, the vectors require a selectable marker which helps in identifying and eliminating non-transformants and selectively permitting the growth of the transformants. For example, genes encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline, etc. used a selectable marker. Cloning sites. In order to link the aligned DNA, the vector needs to have very few, preferably single recognition sites for the commonly used restriction enzymes. Such enzymes may be EcoR1, Hind3, Sal1, etc. Types of vector. Some commonly used vectors are plasmid, PBr322 and TA plasmid. As we know, plasmids are extrachromosomal, self-replicating, double-stranded circular DNA molecule found in the bacterial cells. These plasmids contain genetic information for their own replication. PBr322 plasmid is a reconstructed plasmid and most widely used is cloning vector. It contains 4361 base pairs. In PBr, P denotes plasmid. B and R respectively the names of scientists Bolivar and Rodriguez who developed this plasmid. The number 322 is the number of plasmid developed from their laboratory. It contains ampicillin resistance and tetracycline resistance genes and recognition sites for several restriction enzymes such as Hind3, EcoR1, BOMH1, Sal1, etc. ROP codes for the proteins involved in the replication of plasmid. Next one is TA plasmid. TA plasmid is found in Agrobacterium tumefaciens. It is a soil inhibiting bacterium. This bacteria responsible for inducing tumors in several dicot plants. The plasmid carries transfer gene which help to transfer tDNA from one bacterium to another bacterial or plant cell. It has ONC gene for oncogenicity, which means tumor inducing, origin gene for replication, and INC gene for incompatibility. tDNA of TA plasmid is stably integrated with plant DNA. Agrobacterium plasmids have been used for introduction of genes of desirable traits into plants. Next one is competent host for transformation with recombinant DNA. The propagation of the recombinant DNA molecules must occur inside a living system or host. Many types of host cells are available for gene cloning, which includes E. coli, yeast, animal or plant cells. The type of host cell depends upon the cloning experiment. E. coli is the most widely used host organism because its genetic makeup has been extensively studied. It is easy to handle and grow, can accept a range of vectors, and has also been studied for safety. One more important feature of E. coli to be preferred as a host cell is that under optimal growing condition, the cells divide every 20 minutes. Now let's see how the transformation takes place in the host cell. DNA is being hydrophilic molecule, so to pass it through cell membrane, the bacterial cells treated with a specific concentration of a divalent cation such as calcium. Recombinant DNA can be forced into such cells by incubating the cells with recombinant DNA on ice, followed by placing them briefly at 42 degrees centigrade and then putting them back on ice. This enables bacteria to take up the recombinant DNA. For the expression of eukaryotic proteins, eukaryotic cells are preferred because to produce a functionally active protein, it should fold properly, and post-translational modifications should also occur, which is not possible by prokaryotic cells. These are the basic tools required for genetic engineering process.